So this is my presentation on horizontal gene transfer, specifically that between bacteria. Okay, so basically the idea behind horizontal gene transfer is that bacteria can swap DNA, and there are a few definitions for this type of thing. One is that it's a process by which organisms transfer genetic material to organisms which are not their offspring. So normally us mammals, we'd have mom and dad, and then we transfer our genetic material to our children. But horizontal gene transfer can be defined as a process by which you share genetic information that is not passed on through reproduction. More specifically, getting into what actually happens, it's the mobile nature of gene islands, which are transmitted between bacteria via plasmids, phages, and other methods which create potential for acquired virulence in previously innocuous microbes. So breaking that down a little bit, what it means is that these guys can, through a variety of mechanisms, share pieces of their DNA with each other, and the part about creating potential for acquired virulence means that they can go from being a very nice bacteria to a very bad bacteria because they suddenly know something, they get a piece of DNA that tells them how to do something that's harmful to the human body. So, for those of you who've seen The Matrix, it's kind of like Neo when he gets Kung Fu downloaded into his head, which is what's happening right now, is that the bacteria get the DNA and they're like, I know how to defeat the body's immune system now. And that's really cool. Approximately 20% of the genome of the bacteria E. coli, which you probably know about, probably originated from horizontal gene transfer, which is just bacteria sharing their DNA like this. They can use horizontal gene transfer to figure out ways to resist antibiotics. So if you have heard of those instances where, yeah, they've got these superbugs in hospitals that are super resistant to antibiotics, that's probably thanks to horizontal gene transfer. A couple examples of bacteria that have this are Shigella flexneri, which causes diarrhea, and Staphylococcus aureus, which you probably know causes staph infections. So there are three ways that horizontal gene transfer can happen. It can happen through transformation, transduction, and conjugation. So transformation is basically the bacteria absorbing DNA from other dead bacteria in its environment. It really only absorbs short pieces of DNA, and only certain types of bacteria can do it. So if you don't have the right kind of envelope to take in naked pieces of DNA, then it's not going to work for you. Transduction is sort of like a happy accident for bacteria sometimes. Strands of DNA get carried through a bacteriophage, and that's this creepy looking guy here, and we'll explain more how that happens, but basically he sucks up bacterial DNA and goes around to other bacteria and puts it back in them. So that only happens with really closely related bacteria because he needs to hold on to little ports on the outside of the bacteria in order to inject the DNA. So that's also a limitation for these two. Conjugation is the most common. That happens with plasmids and transposons, and it transmits the most amount of DNA at any one time. So that's the kind of the big scary head honcho of all of them. So we're going to talk about transformation first, which is when bacteria absorb DNA from their environment, from other dead bacteria. So we really first started learning about transformation with Griffith's experiment, which was an experiment by a researcher named Frederick Griffith. He was looking for a vaccine to pneumonia, which is caused by the bacteria Diplococcus pneumonia. And there are basically two strains. You have a rough strain, which is not virulent, and a smooth strain, which is virulent. And what makes the smooth strain virulent, which virulent means that it can take over your immune system and do nasty things to you, what makes it virulent is that it's got this really smooth polysaccharide capsule around it, sort of protecting it. The non-virulent strain does not have that polysaccharide capsule. So he injected a bunch of mice with the different strains. He's got, this mouse gets the non-deadly strain, it lives. Makes sense. This mouse gets the deadly strain, it dies. That makes sense. So when you kill the deadly strain, it can't do anything, so yeah, the mouse lives, it's okay. But when you mix the harmless strain with the one that should be harmless because it's dead, you kill the mouse. And that was the sticker for him. He was like, so how does that happen? He called it a transforming principle, where somehow just through contact with the heat-killed bacteria, these smooth, harmless strains were getting the ability to defeat the body. And that was before we even knew what DNA did, so kudos to Griffith, because that was a bit ahead of his time. So what happens is the bacteria are basically taking the DNA of the dead bacteria that are around. They're sort of absorbing it and using that. So if you wanted to, like, a memory marker, it's sort of like stealing from other bacteria. You know, the other bacteria, they've got this DNA, and you just kind of take it and use it to further your own survival. And some examples of bacteria that can do this, only a certain type can, are Streptococcus, Staphylococcus, Haemophilus, and that causes pneumonia and meningitis, and Pseudomonas, which causes infections in hospitals often, probably thanks to horizontal gene transfer. So next, we've got the really cool one with our little bacteriophage friend. This is transduction. The way to remember that is transduction is transportation, so it's going across different bacteria. Basically, a bacteriophage, which is this little guy, he's gonna pick up, basically the way they work is they have their DNA, and it's kind of like a virus. They take their DNA, they put it in, and then they replicate it and they burst out. But with transduction, something happens along the way where, okay, you got all these guys replicating inside the bacteria, and one of them gets the DNA from the bacteria. And it just sort of spreads it all around to other bacteria instead of actually causing DNA, even DNA that causes the bacteriophage to keep replicating. So it's kind of like this weird, oops, I gave you the wrong DNA and it happens to be an advantage to you sort of thing. So I have an example, a couple animations up to show, because it's kind of hard to explain otherwise. Here we go. Alright. Okay. So this is generalized transduction. You've got your little bacteriophage, right? He has the bad DNA, the one that replicates stuff. And here's your donor bacterium. And you see the little bumps on there? That's what I was talking about with it needs something to hold on to, that they have to be from the same family, otherwise it's not really going to work. Okay, so we can start that. And he's just coming on here. I'm going to simplify what's happening a little bit because the whole thing's actually really complicated with vocabulary and everything. So it's just inserting the DNA, which is what's going to turn into more of these inside the bacteria later, and it breaks down the DNA in here, and this is generalized transduction. There are two types, generalized and specific. In generalized, it accidentally forms around some of that DNA. So, oops, that guy is not like his brothers. He's different. He's not dangerous. So he's coming out, and he lands on the cousin of that bacteria that just died. They've got the same ports, so he can jump on there, and what happens is he inserts the wrong DNA, the DNA he accidentally formed around, and that goes into the donor bacterium. So, whoopee, instead of dying and bursting with bacteriophages, you got a generous donation of genetic code. So, we also have specialized transduction, which... gotta scroll. Okay, cool. Because I don't have a scroll bar. Okay. So we also have specialized transduction. So in specialized transduction, a bacteriophage comes on and it latches onto the bacteria, and you know, it's the same thing as we saw in the start of generalized transduction, yada yada yada. It's just a bacteriophage replicating. So let's go. But here, it inserts into the bacterial genome, and at this point it's called a prophage. And you know, the bacteria, okay, it goes through its normal life cycle, it replicates, it has a daughter cell, and that prophage excises itself, and sometimes it does that wrong, and it takes a bit of the bacteria with it. So that bacterial DNA becomes part of what the bacteriophage replicates itself as. And it's called specialized transduction because it's got that specific gene in there that they're all carrying everywhere. So now they go give those genes to other bacteria. So when that bacteria gets a little blue piece of DNA that was from the other bacteria inserted into its genome, as you can see happening, all of a sudden it's got DNA from another bacteria in there. And that's specialized transduction. So that's the simplified version of what's going on there. So, what you remember is there's generalized and specific. And last, we've got the big head honcho conjugation. So basically, these, in conjugation, the cells need to maintain contact through a conjugative pilus, and that's this thing here, and they will transfer DNA typically through plasmids, which are like rings of DNA. That means you can see they can just send through the pilus there to swap with each other. Sometimes it happens with transposons, and transposons are like little genes of DNA, kind of like we have, except they're surrounded by enzyme markers, and they can just kind of hop out and leave and go somewhere else and reinsert themselves into another strand of DNA, whether that be inside the same chromosome or into two entirely different bacteria. So that's pretty cool, is that these jumping genes can just kind of hop from bacteria to bacteria, and that requires a conjugative process. So conjugation is where they come together and they start sharing information. Those are my references. And last slide to leave you with stuff to think about, because I don't think that anybody could ask me any questions right now. But thanks for watching.